EDU-KV, let's come together to learn.
Hello friends, today we shall discuss about the third chapter of class 12th biology, that is human reproduction. In this video we will study about the gametogenesis, spermatogenesis, spermatogenesis and hormones, structure of sperm and oogenesis. And this is the third video in continuation of the previous two videos, and in this video we will be able to learn about the gametogenesis and oogenesis. Okay friends, let's start one by one. First thing which comes to our mind about the gametogenesis, the primary sex organ, the testis in the male. The primary sex organ is the testis in the male. Okay, now the process of formation of haploid gamete from diploid germ cells in the gonad is known as gametogenesis. Second point is that in testis, the immature male germ cells or the spermatogonia produce sperms by spermatogenesis that begins at puberty. And these primary sex organ, specifically testis and ovary, they produce sperm and ovum. Male, in the case of male, spermatogenesis, and it is produced by the help of spermatogonia and it begins at the puberty in the case of male. While in case of females, oogenesis is the process through which gametes are producing, that is specifically ovum is being produced, and then it starts at the embryonic stage. That means oogenesis will start at the embryonic stage. Now the next point is that the spermatogonia, singular spermatogonium, present on the inside wall of seminiferous tubules, which multiply by mitotic division and increase in numbers. Okay, and the process of formation of haploid male gamete sperm in seminiferous tubules is termed as spermatogenesis. Etymologically, the whole word spermatogenesis made up of two different words. First one is the spermato, another one is the genesis. Spermato means formation of sperm and the formation or the genesis means production. Okay, the process of formation of haploid male gamete, that is sperm, in seminiferous tubules of testis is known as spermatogenesis. As I have already told you that the inner wall of the seminiferous tubule contain two different type of germ cells, and the germ cells are commonly called as spermatogonial cells. First one is the germ cell, that is spermatogonial cells, and second one is the Sertoli cells. These are the two different type of cells that are present in the inner wall of the seminiferous tubules. Germ cells divide and develops into sperm. Sertoli cells nourishes the developing embryo. Actually, germ cells divides and develop into the sperm, while the function of the Sertoli cell is to provide the nourishment to the developing sperm. The spermatogenesis actually takes place in two stages. First one is the spermatidogenesis and the spermiogenesis. Spermatidogenesis or spermiogenesis means the formation of spermatids, initial formation of spermatids, and then spermiogenesis we will see later on. Apart from this one, this phase is there that the diploid primary spermatocyte undergoes first meiosis and resulting in the formation of two haploid cells, and they are known as secondary spermatocyte having 23 chromosomes, and this latter undergoes second meiotic division to form 4 equal haploid spermatids. Now the next process is spermiogenesis. What in the previous slide I have told you, two different processes. First one is the spermatidogenesis, another one is the spermiogenesis. Spermatidogenesis is the formation of spermatids. That means there are 4 equal haploid spermatids are there. Another one is the spermiogenesis. It is inactive non-motile spermatids are transformed into active motile spermatozoa. The process is known as spermiogenesis. Spermiogenesis. Spermiogenesis is the process of conversion of inactive non-motile spermatids. Actually, initially spermatids are inactive and they are transformed into active motile spermatozoa, and the process is known as spermiogenesis. After spermiogenesis, sperm had embedded in Sertoli cells. Let's take a diagram of seminiferous tubule. Inside the seminiferous tubule, actually what happens, the sperm's head embedded in the Sertoli cells and it released from the seminiferous tubules, and the process is known as spermiation. So students, total 3 different processes we have already studied. First one is the spermatidogenesis, that means formation of spermatids. Second one is the spermiogenesis, conversion of inactive non-motile spermatids into the active motile spermatozoa, that is the sperm. And the third one is the spermiation. That means release of sperm which already embedded in the Sertoli cells from seminiferous tubules, and the process is known as spermiation. Now, as all of you know that spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone, that is GnRH, gonadotropin releasing hormones. Now, spermatogenesis is directly related with our hormones. First one is the GnRH, that is gonadotropin releasing hormone. This is the hypothalamic hormone. It releases from the hypothalamus. So it is the super master gland. It can be called it as super master gland. Okay. Now, all of you know master gland is anterior pituitary or the simply pituitary gland. GnRH, that is gonadotropin releasing hormone, stimulates the anterior pituitary gland to produce the gonadotropin, and these gonadotropin will act on the 2 different type of hormones, and it initiates or to release the luteinizing hormone and the follicle stimulating hormone. And this luteinizing hormones actually works on Leydig cells to produce the androgens. Here androgen is the common term. Specifically, a hormone is known as testosterone produced by Leydig cells. Okay. Now the follicle stimulating hormone, it acts on the Sertoli cells. As I have already told you, Sertoli cells are those cells which acts or which helps in the nourishment of the developing sperm cells. Okay. Now these androgens or testosterone stimulates the spermatogenesis, and this Sertoli cells secretes the factors for spermiogenesis. Okay, these are the 2 different processes, spermatogenesis and spermiogenesis, which I have already discussed. Now after these this conversion of spermiogenesis and the spermatogenesis, actually when we study, let us examine the structure of the sperm in a microscopic structure composed of head, neck and middle piece or tail. Actually this is the structure of sperm. Sperm has, this one is the head portion, there is a small neck portion and then middle piece, and this one is the tail. Okay. Now covering this whole structure, that is the plasma membrane, envelopes the whole body of sperm. Plasma membrane covers the whole body of the sperm, and then the sperm head contains an elongate haploid nucleus. There is a haploid nucleus is present on the head region, and the anterior portion of which is covered by a cap like structure that is known as acrosome, and this acrosome is filled with hydrolytic enzymes. It is filled with hydrolytic enzymes that helps in the fertilization of the ovum, and the middle piece possesses numerous mitochondria. This middle piece contains numerous mitochondria. As all of you know that the function of the mitochondria is to provide energy for swimming. Okay, it provides energy for swimming of the sperms and then facilitate the sperm motility essential for fertilization. And the human male ejaculates around 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size, and at least 40% of them must show vigorous motility. Okay, now this is the point that after the sperm structure, plasma membrane envelops entire body. First portion is the head. It is the elongated haploid nucleus. Anterior cap like acrosome is present. It has hydrolytic enzyme, specifically hyaluronidase. It is derived from Golgi complex during division, fertilization of ovum. Then neck portion consists connecting head and the middle piece. Middle piece consists of many mitochondria, produce energy for the movement of the tail, that is motility, and tail is the long slender vibration. Around 200 to 300 million sperms releases during one ejaculation. Around 60% must have normal shape, size, and 40% have motility. Okay, now apart from this sperm structure, the next process which comes to our mind is the oogenesis. Oogenesis is the process of formation of mature female gamete. It is the process of formation of mature female gamete. Here female gamete is the ovum, and it developed from the oogonial cell, and the oogonial cells are diploid in nature and they are present in the ovary, and the process is known as oogenesis. And the process of oogenesis begins at embryonic development. That means this is the must point, which please remember that the oogenesis begins at embryonic stage. Okay, now first thing is the oogonia, which is 2n diploid in structure, that is gamete mother cell, and at birth it may be millions in fetal ovary. Okay, now during primary oocyte, after oogonia will get converted into primary oocyte after meiosis, that is reduction division, and this primary oocyte will cease at a stage of prophase 1. It will cease at a stage of prophase 1 of the meiotic division, and it is the temporary arrested in this stage. This is to be kept in your mind that during primary oocyte formation, the whole process ceases or arrested during this stage in the prophase 1 of the meiotic division. After that, this primary oocyte will get converted into primary follicle, and this primary oocyte consists of primary oocyte and granulosa. This is the primary oocyte and granulosa cells. Covering will take place, and many follicles degenerate from birth to puberty. Around 10 to 13 years, after the span of 10 to 13 years, many follicles degenerate. Now minimum 60 to 80,000 in each ovary, these primary follicle cells are present. Okay, after that these primary follicles will get converted into secondary follicles after the conversion of deposition of more granulosa cells, and this primary follicle surrounded by more granulosa cells, and it will form the theca. Okay, and it will form the secondary follicles. After that, these secondary follicles will get converted into tertiary follicles. Again, secondary follicles, when filled with fluid filled cavity, that is known as antrum, fluid filled cavity is formed, and this is the external thecal layer. It is known as theca interna, vascular layer, and theca externa, that is fibrous. theca interna is 10 to 15 layers thick. It is a part of membrane granulosa. And then primary oocyte, this structure, primary oocyte, which is 2 in number, and it is present inside this follicle, and its size will increases, and first meiotic division, that is unequal large haploid secondary oocyte and first polar body, will take place. Up to this stage, actually what happens, primary oocyte, which is diploid in nature, arrested in the prophase 1 of the meiotic division. Initially its size will increases, and then this prophase or the meiotic division will get completed here, and there is unequal division will take place with a large haploid secondary oocyte. Primary oocyte will get converted into secondary oocyte, and another one is the first polar body. Okay, first polar body. After that, this tertiary follicle will get converted into secondary oocyte. Now secondary oocyte will form, and this secondary oocyte retain the nutrient rich cytoplasm of primary oocyte, and then this secondary oocyte along with follicular layer, it will form the tertiary follicle. Here now secondary oocyte will form, and this is the follicular layer, and now this structure is known as tertiary follicles, and this tertiary follicle will ultimately get converted into Graafian follicle. And now this secondary oocyte, that is ovum, and zona pellucida, that is the membrane surrounding the secondary oocyte, and the Graafian follicle ruptures at last and it releases the ovum. This is the whole process of oogenesis. What happens in the oogenesis? Initially mother cell or oogenesis starts from the haploid gamete, formation of the haploid gamete, that is the ovum in the follicles of the ovary, and the process is known as the oogenesis. Initially oogonium is there, and is oogonium, after mitosis it will form oogonia, and then cell growth will take place. It will form the primary oocyte, and this primary oocyte is 2n in number, that is diploid, and then meiosis 1 arrested at the stage of prophase 1, reduction division, and then afterwards the secondary oocyte and the first polar body will form. What happens? Actually oogonia enters into the meiosis 1, proceeds towards prophase 1 and gets suspended and form the primary oocyte during puberty, and the primary oocyte restarts its first meiotic division during puberty. Okay, after attainment of the puberty, the child or the female individual, the primary oocyte restarts its first meiotic division, and now the first meiotic division will get completed there, and the oogenesis takes place by the 3 stages. First one is the multiplicative phase, second one is the growth phase, and the third one is the maturation phase. As you know, multiplication phase, this is the phase in which the certain primary germ cells, that is the large size and nucleus of germinal epithelium lining ovary, undergo rapid mitotic division. It results in formation of group of diploid egg mother cell, that is the oogonia, what I have told you. This is the flow chart of the oogenesis, and each group of cells form a round mass called egg nest or egg cell. Okay, now is the growth phase. Second phase is the growth phase. It is the longest duration, that is it around 12 to 13 years. One of the diploid cell, that is the oogonia, undergoes growth, increasing in the cytoplasm, and accumulation of yolk transformed into enlarged oogonia called primary oocyte. Okay, here you can easily very well understood how the size of the primary oocyte will increase. The chromosome number remains same, that is 2n, but after formation of primary oocyte, the other oogonia forms single layered follicular epithelium around the primary oocyte. Primary oocyte is there, and then other oogonia forms single layered follicular epithelium, and this primary follicle surrounded by more granulosa cells will form secondary follicle, and this secondary follicle full fluid filled antral cavity. Antral cavity means there is a cavity present inside this secondary follicle, and that is known as and it will become the tertiary follicle. After formation of the antral cavity, that is antrum, it will get converted into tertiary follicle, and this tertiary follicle will form ultimately Graafian follicle. The third stage in the formation of oogenesis is the maturation phase. A fully grown primary oocyte, that is which is a 2n in number, that means diploid, undergoes first meiotic division, results in the formation of two unequal sized haploid cells. One is this one, this is the secondary oocyte, and haploid polar body, that is polocyte, polar body is there, and the secondary oocyte again undergoes meiotic division, second meiotic division, to form a large ooted and the small second polar body. Now you can very well understood here, two different small polar bodies are formed and they will ultimately degenerate, and now the second oocyte which is present in new membrane, that is known as zona pellucida, and it will form the Graafian follicle, and the first polar body also undergoes equal division to form the two cells. Okay, it will also form the two cells. Now apart from this one, this stage, thus during oogenesis, four cells are produced. Among them, one is functional ooted, another one three non-functional polar bodies. Three non-functional polar bodies are formed. Actually, first one is the first polar body. This is the first polar body, and this is the secondary oocyte, and this first polar body again will form two polar bodies and it will form one polar body. Okay, here total three polar bodies are formed and a functional otid is formed. This is the functional otid, and all these three are polar bodies, non-functional polar bodies, and the otid with very little change becomes an ovum, and this otid will become ovum. At last it will form ovum. Okay. Okay, thank you very much.
Please subscribe, like and share the channel.